Hi Nat5s, so we're going to look at transport across the cell membrane. That's the key area. In particular, I want to look at the composition of the cell membrane. In fact, all membranes in a cell. So the membrane, which you drew as basically just a line, it obviously has to be more complete than that, because, and a lot more complicated, because you know that the membrane controls what enters and exits a cell. So if it was literally just made of one thing, how does it control what goes in and out? So there are actually two bits, okay, something called phospholipids and proteins, okay, and you're going to just need to know those terms. And what they're arranged in is something called a fluid mosaic. So we're going to look at that, break it down into two parts. What do we mean by fluid and what do we mean by mosaic? Okay, so the fluid bit is actually made of these things called phospholipids. And phospholipids are quite strange molecules. They're what's called a hybrid molecule because they have two ends in terms of how they behave. So if this is the phospholipid, we normally just draw it like, oh, sorry, wait a minute, get my pen up. We normally just draw it as a circle with two lines coming down. But this is actually quite a complicated chemical. This circle bit at the top, so this bit up here, it really loves water. It's really happy to be near anything that's watery. And that's basically most of you. So the outside of a cell will be watery and the inside of a cell will be watery. But then we've also got this bit down here, these legs or tails, and these bits are hydrophobic, meaning that they hate water. If you have a phobia, you have a fear, a hatred of something. So this is hydrophobic. So this bit hates being near water, so it doesn't like being directly to the outside of a cell, or indeed pointing directly into the cytoplasm. So what you end up with is something called a bilayer. So bi, like bicycle, means two. So what you actually end up with is something like this. This has been kind of opened up to see it. So here's all the heads on the outside, which are really quite happy to be next to liquidy stuff on the outside, water. The inside bits, the tails, stick next to another set of tails. So you've basically got head to tail and then another tail to another head. And that just keeps going. And then you end up with this huge big thing. This, I say, is peeled open just to see it. So on the inside of the cell, this would be the inside bit here. All the heads are happy because they're next to the cytoplasm, which is all watery. And on the outside of the cell, we've got another layer of heads, which are also happy because they're next to water as well. So you just need to know how these shapes arrange, with the circles on the outside and the inside and the tails all the way through the middle. Okay. So the reason that's fluid is because they can move. So if you have your kind of layer of these little heads to tails, so these can move around. So like this one can kind of move around inside this layer, and this one can move around inside this layer, but they can't flip from one side to another. They can't do that, but they can move around inside it. So this is why it's the fluid part of a fluid mosaic. Right, part two, the mosaic. So this is made of proteins. So if you think of what a mosaic is basically something that's made up of lots of little pieces. And if you also think about it, you can have lots of different things that you want to happen inside a membrane. So you might have proteins that are carriers, need to pick things up. You might have things that are signals that say, hello, I'm a liver cell, or hi, I'm a nerve cell. They do lots and lots of different jobs. So if you think again of a mosaic, you can make every single picture you could want as long as you have all the different colours. So if you think of proteins as being different colours, you can make millions of different mosaics. So you can make millions of different cell membranes. You can make a cell membrane on your heart cell look very different from a cell membrane on your gut cell. Okay, it just depends on what ones you put in. But that means that you have quite a complicated thing now. It's not just a black line, it's a really complicated thing, and quite a clever thing. So different cells, because they have different proteins, can do different jobs. So when we talk about the membrane, and this is going to be really important for this key area, we talk about being selectively permeable. So the term permeable, I'm not sure if you will have seen it. You might have seen it about fancy jackets and things. They sometimes say things on them saying it is impermeable to wind, meaning wind cannot get through it. They might also say that it's a permeable membrane so that the water can get through. So if you sweat, it will be taken away quite quickly. So you might see things, might have seen stuff there. This is actually a picture of trying to show that this is a gravel that you might want to put on your driveway because it's permeable to water, so it lets the water through so it doesn't sit on the drive. It's the only place I think you might have seen them though. So the term permeable is how much is let through. So if something is selectively permeable, you pick and choose what goes through. Well, that's the definition of a membrane, controls what goes in and out. So that's what we mean when we say selectively permeable. So if you see those words, don't freak out. That's what they mean. So what will happen with that is that a cell can actually decide to have different concentrations of things inside the cell and outside the cell. So it can concentrate stuff inside that it wants to be there, and it can stop things coming in that it doesn't want to come in. And that's going to be important when we go in to talk about how membranes work. So this would be a kind of proper picture now of a membrane. So instead of you just doing here's a cell with a nucleus in the inside and that's the cell membrane, this bit here actually looks like this now. Okay, so we've got our phospholipids with their heads always on the inside and the outside, and the tails always pointing into the middle, and then we have proteins that are slotted in to make the mosaic part of it. So you've got this one here is just sitting across half of it. It's called a peripheral protein because of that. And then we've got a channel protein, so this is basically making a hole straight through the membrane, and this channel will be just one specific shape, so it might be exactly the right shape for glucose, which means glucose will get through but nothing else will. And then we've got special carrier proteins, which we're going to talk about a little bit more further on, and these ones, you'll notice this looks closed at the moment. This bit here looks like nothing would get through, because to open it you need to use energy. So these can pull things across, things that wouldn't normally get across the membrane, they can yank them across. This is another type of protein. Don't need to worry about the type of this one, but this would be something that would be a receptor or would be a signal to say that you're you, which is kind of important for your immune response. Okay, so just to say that one of the experiments that we might do to prove this, and if we were in school we would definitely do this one, you take something like red cabbage, and in a red cabbage cell, if we could wreck the cell membrane and the vacuole, then all the colour would leak out. So what you can do to prove that we have phospholipids and proteins, we can take a high temperature, which will basically cook the protein, change its shape, so things would leak out. We can use ethanol, which dissolves lipids, so that would get rid of the phospholipids, and we can use acid, which also damages protein. So we can temperature and acid are to check to see if there's protein there, and the ethanol is to check to see if there's lipids there. So what we would do is take some boiling tubes, put some water in two of them, ethanol in another, and acid in another. We'd get little discs of red cabbage, rinse them in cold water first before we put them in, just to make sure that they didn't have any liquid colour on the outside of them. And then we'd take one of our test tubes, put them into 50 degrees, and then we'd wait for 40 minutes and then take them out. And what would happen is, if this was the room temperature one, so this one's just been left at room temp, and this one has been up at 50 degrees C. Okay, in this one you might have a little bit of redness around the outside, but very, very little. It won't have leaked much at all. This one, however, should have gone pretty pink. Like, most of this will have coloured in pink. The ethanol will also have leaked out loads of colour, and the acid will leak out loads of colour. So these two here prove that proteins must have been part of the membrane, because when you wreck the proteins, bits come out of the membrane. The colour that was being held inside membranes comes out. And this one proves that you have lipids. Okay? So you might be asked some questions about experiments. I'm not going to go into these in detail. I just want you to think about these kind of questions. So what variables should have been controlled? So if you think about that experiment, how many things would I actually have had to tell you measure this or do this for a set time, or think about what you would actually have had to control to make sure that it was fair. Which test tubes in there are actually controlled? So the thing that you could compare the other ones to say, yep, that definitely made a difference. And this is one of the things we might use. Egg white is pure protein. It's made of something called albumin. So if we wanted to use this as a control, where would you put it? Like, where would that actually be put to prove that protein was involved? And this is something that would be nice to think about. It's all very well saying that went a bit pink, but is there a way to get a more quantitative data set? And by quantitative, I mean, could you have some numbers in there? Can you think of a way where you might actually get some numbers in this one? There is quite a bit to go through in that one in terms of the new terms, but pretty much what we're looking for is phospholipids, proteins, fluid mosaic. Make sure you're happy with those terms. Okay.